And you got dropped.
Yeah, I know I had to go to the alley.
Okay. Yeah, apparently you got dropped from all your classes.
Yeah. There's some like crazy stuff.
Yeah, there's been a lot of that recently. So these are, again, the learning goals that the book says. So what kind of interactions occur between molecules? Can anybody name those, like general chem stuff? See, yeah, you all know all this already, I'm sure, so we don't have to spend hours and hours talking about van der Waals forces, hydrogen bonds, dipoles, and ionic, things like that, right? So why water is a good medium for life, do you guys know? Yeah, it's a great solvent, right? So it's good solvent. And you guys ever heard of hydrophobic properties? Like, yeah, we'll talk about those more. And then you guys remember weak acids and weak bases? So how to calculate pHs and concentrations and stuff like that with that, what's that, Henderson-Hasselbalch equation? And then your book, I don't think, talks much about bicarb, carbon dioxide, equilibrium in the body, but I'll cover that a little bit. Since probably half the class is CLS, that'll be on those board exams also. And it's good to know it's pretty easy. So life evolved in water. One of the reasons is UV protection. So what does UV do to DNA? Do you know?
Thymine dimers?
Yeah, you can get thymine dimers. UV is, what does it do though? It excites electrons, right? And it's going to cause chemical reactions. So organisms typically contain 70 to 90% of water. Chemical reactions occur in this aqueous milieu, right? So it's that water that allows these chemical reactions to occur. And water is a critical determinant of the structures and functions of proteins. So it's the water helps shape how those proteins fold, basically. And a lot of that has to do with the hydrogen bonds, van der Waals forces, and hydrophobic interactions. So, and it also functions of proteins, nucleic acids, and membranes. So, and all of these, right, we talked about last time, they're polymers pretty much, right? So do you know what kind of reaction is involved at forming those polymers? Because that involves water too, right? What?
Dehydration. Yeah,
dehydration reaction. You remove water from those two chemicals to form that polymer. So your the bonds between the phosphate and the hydroxyl to form a nucleic acid extension, that's a dehydration. You remove water between those two. Also the peptide bonds, when you form those, you get water as a byproduct out of that also. So we all know this general chemistry stuff, right? Octet rule dictates that there are 4 electron pairs around an oxygen atom in water. So that means 2 of these pairs are covalently linked with the hydrogen atom, and you have two free pairs, right? And that's what gives it that distorted tetrahedral shape, right? The 2 remaining pairs remain non-bonding. The lone pairs in the water geometry, like I just said, is a distorted tetrahedron. And the electronegativity of the oxygen atom induces a net dipole moment. Because you got partial positive charge, right, on both the hydrogens. Partial negative, and I forget the little squiggly or whatever for the partial negative on the oxygen. Something like that. I'm a terrible artist, so please forgive me. And this is the figure from your book. I guess I didn't have to draw it if I just go to the next slide. So because of this bent shape, you get partial, it's polar basically, right? Plus you have these electrons out here and you have hydrogens, so that makes water available to do what? Form hydrogen bonds. So how many hydrogen bonds can one, water molecule form?
2.
No.
4.
4, yes. You can accept 2 hydrogen bonds with the lone pairs and then donate 2 be the hydrogen donator for 2. So it's 4 hydrogen bonds 1 water molecule can form at once. In fact, the crystalline form of water is basically you have 4 hydrogen bonds formed. So because you're forming 4 hydrogen bonds at one time in crystalline water is, what's the property of ice, one of the properties of ice that makes life possible here on the planet? Yeah, ice, the crystalline form of water is less dense than the liquid form, so it floats and kind of protects that liquid form from, freezing all the way through. So you can get, life gets preserved in the liquid form of water underneath. Does that make sense? And I'm probably jumping around in this lecture because I guarantee that's later on in this PowerPoint too, so please forgive me. So we were talking about hydrogen bonds. Somebody want to define that for me? So basically strong dipole-dipolar charge dipole interactions that arises between an acid, a proton donor, and a base or a proton acceptor. And they're typically 4 to 6 kilojoules per mole for bonds of neutral atoms, or with charged atoms 6 to 10. Typically involves 2 electronegative atoms, nitrogen and oxygen most frequently. You can also hydrogen bond with fluorine also. And hydrogen bonds are the strongest when the bonded molecules are oriented to maximize the electrostatic interaction, right? So that's the crystalline form. You form 4, and there's a lot of empty space there, which is why water is less dense. So. And ideally, the 3 atoms are in a line, and that's what this figure shows. You get a strong hydrogen bond when these 3 atoms will be in a straight line. It's a little bit weaker if they're not perfectly angled. So, and you'll see that in protein structures. You'll have some structures that are hydrogen bonded a lot stronger together, and not just protein, but nucleic acids also, the way that, you'll base pair with each other. So. And water can serve as both a hydrogen donor and an acceptor, and we can form almost 4 hydrogen bonds per water molecule, which gives water its high boiling point compared to other, hydrogen bonding atoms. Anomalous high melting point, usually large surface tension. And what are all these why do you get these from water? What's that property called? It's because of its strong intramolecular forces because of the hydrogen bonding, right? So hydrogen bonding in water is cooperative, and bonds between neighboring molecules are weak, 20 kilojoules per mole, relative to the OH covalent bonds, which are about 420 kilojoules per mole. So meaning that when you form these hydrogen bonds, they're going to be transient because they're weak, whereas covalent bonds typically are more permanent. They're not as transient. So you'll have, in liquid form especially, as you increase the energy of water, you'll have shorter and shorter hydrogen bond, not bond length, but time-wise, molecules forming hydrogen bonds with each other. So, and this shows you bond length as a factor. You saw the difference in energy of the bonds. The covalent bond's much shorter than a hydrogen bond. And you guys remember that from general chem. The shorter the bond length, the stronger the bond, right? So water has many different crystal forms, and this hexagonal ice is the most common. And that forms a regular lattice structure and thus has low entropy. So it's got a lot of order, right? So it has low entropy. And hexagonal ice contains more hydrogen bonds per water molecule, thus ice has a lower density than liquid water. I think liquid water has on average about 2.3 hydrogen bonds per water molecule versus the crystal is 4 hydrogen bonds per water molecule. And that's because the liquid is so much more transient because it's in motion all the time. And half the hydrogen bonds aren't going to be that perfect angle, so they're a lot weaker also. So. And this is the figure from your book showing you the crystalline structure of water, forming 4 hydrogen bonds per each water molecule. So water is also a good solvent for charged and polar substances, which include amino acids and some peptides, small alcohols, and carbohydrates. It's a poor solvent for nonpolar substances such as nonpolar gases, aromatic moieties and aliphatic chains. So like water forms drops in the air, right? So why does it do that when it's interacting with nonpolar gases such as their atmosphere? Why does it normally form a drop?
Lower surface tension.
No, it's not because of lower surface tension, but surface, I think you're thinking not surface tension, but surface area, right?
Surface area.
Yeah. So it reduces the amount of surface area of the water molecules that have to interact with the air. So, and you'll see the same thing with hydrophobic forces that are, phenomena. That's what governs folding of like oil. If you put oil in the water, it forms little droplets also. It's trying to reduce that surface area, right? Of how much of that oil or how much water is going to be exposed to the oil. And this is a figure from your book. So things with lots of polar and hydrogen bond capability are going to be good solutes in water, such as sugars, like glucose, glycine. Anybody know what kind of ion this is? Well, yeah, there's an amine group and a carboxylic acid, but what is it when you have this on one molecule? One positive charge, one negative charge.
Oh,
It's ionic, yeah, but there's no net charge, right?
neutral.
But it's called a Zwitter ion. ZWE, I don't know. I got it later on, I think, at least on the next chapter. I'm a bad speller. I could try. It's like, I think that's how you spell it. I'm a small writer too. And it's a bad color I picked, I think. So, huh? Sorry. I could do it in black again. Normally these don't work. Okay, Zwitter ion. So this would be a Zwitter ion. No net charge, but you do have a positive and negative charge on the molecule. Aspartate, lots of charge, so it's good and soluble. Lactate and glycerol, lots of alcohols, glycerols, alcohol that's soluble, polar groups, and they have the polar groups highlighted. Whereas non-polar groups, you have a wax. Anybody know what a wax is? What kind of bond is this? No, so like if you look at this is a fatty acid, so this is the carboxylic acid group on a fatty acid, and this is a long chain fatty alcohol. So you have an ester bond between a fatty acid and an alcohol. That's basically what a wax is. And I think we talk about that in the lipids chapter later on, but just to point it out here. This would be like the head group of the wax, the polar head group, and then the non-polar group. Phenylalanine's an aromatic amino acid, so this is the non-polar group, and then phosphatidylcholine. This is one of the phospholipids you find in the membrane. So you have two fatty acids with ester groups with glycerol, and then a head group that has a phosphoester with choline in between. So water dissolves many salts. It's got a high dielectric constant, which reduces the attraction between oppositely charged ions in a salt crystal. Salt crystals are highly ordered, so water can get in there and reduce the attraction. Right, when they interact with the charges, they're polar. They can help spread that charge out. And start to take apart that salt, which increases the entropy of that system, right? And then it also decreases the heat energy. Well, if you spread that charge, not the heat energy, but the delta H of that system, because you're spreading the charge out. It's a high energy state to have a smaller charge. If you can spread that charge out, like if you solvate it in water, that lowers the energy of that also. And then entropy increases as the ordered crystalline lattice is dissolved. Is that right? So. And this figure showing you that how, and the water will be kind of ordered, which is a decrease in entropy when you order the water around the ions, but still spreading out that charge helps also. But you'll see the water is ordered, the negative side of the water facing the cations, the positive side of the water oriented around the anions. So physics of non-covalent interactions, so we already talked about these. We have ionic, which are charge interactions. Dipole interactions, we'll see that with, dipoles, like, I want to say water, but that's a special case with hydrogen bonds, right? Yeah, water and alcohols have dipole interactions. You can have aldehydes have dipole interactions with each other also, because the dipole on the aldehyde or the ketone. Van der Waals interactions, do you remember those? It's like when the molecules kind of bump into each other. If they're complementary in shape anyways, they'll kind of bump, and that electron cloud kind of gets moved. You get a partial positive and a partial negative, temporary charge interactions due to their closeness. And they're weak interactions, regardless of polarity, and then they can be attractive. And the repulsive part is the steric. If they're not complementary, they can be repulsive to each other also. And then the hydrophobic effect, and this is a complex phenomena associated with the ordering of water molecules around nonpolar substances. And that's the key. The water, when it's got a nonpolar substance mixed with the water's going to be highly ordered around that nonpolar substance. And what happens when the water's highly ordered?
It's a solid.
No, when the water's highly ordered, it's just a decrease in entropy, right? So it's an increase in the free energy of that system. And naturally we all want to get to the lowest state of free energy, right? So that's why you'll want to reduce the amount of surface area of hydrophobic molecules that are going to interact with the water. So examples of non-covalent interactions, we've seen these between two neutral groups. So this is a carbonyl and an alcohol. Two peptide bonds. So this would be the carbonyl in a peptide bond and the amine group in a peptide bond. You'll see a lot of this with protein folding. Ionic interaction attractions, we'll see this with protein foldings also. Repulsion, you'll see that also. And then hydrophobic interactions, they can be very important in ligand binding and folding and all of these represent amino acids. This is a side chain of an amino acid, and so is this. And you can have hydrophobic interactions, going to try to pack together to keep water out, basically, right? And any two atoms in close proximity can, be an example of van der Waals interactions. So the importance of hydrogen bonds, source of unique properties of water, structure and function of proteins. All your secondary structure are formed via hydrogen bonding between the main chain carbons and nitrogens or carbonyls and the nitrogen in your peptide bonds. All your secondary structure is governed by that. Structure and function of DNA. Did you guys go over how DNA base pairs, A's and C's, T's and G's in biology or core biology? So that's all hydrogen bonding, right? Structure and function of polysaccharides, binding of substrates to enzymes, so those side chains in proteins and enzymes are going to interact with their ligands. Binding of hormones to receptors, and matching of mRNA to tRNA. Again, hydrogen bonding between the base pairs. So, more examples of hydrogen bonds, and then more biological relevance of hydrogen bonds. So, this shows you the base pair between adenine and thymine, and that you'd find in DNA. So So van der Waals interactions have two components. This attractive force, London dispersion forces, depends on the polarizability of that electron cloud around the molecule. So when it's polarizable, they'll want to get together and get partial positive and partial negative, and they'll attract. Repulsive forces, or the steric repulsion, depends on the size of the atom. So if you have a big oxygen there, where they're getting in the way, basically, right? It's not very polarizable. Attraction dominates at longer distance, typically between 0.4 and 0.7, and repulsion is going to be dominant at shorter distances. And then there's a minimum energy distance, the van der Waals contact distance, so where you'll have your most attraction there. And these are your van der Waals radii of typical elements. So hydrogen's 0.11 nanometers, 0.15 for oxygen, and so forth. As you can see, as the atoms get larger, the van der Waals distance gets larger. The origin of London dispersion force is the quantum mechanical origin, instantaneously polarization by fluctuating charge distribution. That's kind of what I was saying. It kind of like bump and cause a temporary polarization of the electron cloud. Universal, and it's always attractive. It's stronger and more polarizable molecules, and it's important only at a short range, because otherwise it'll just go back to normal. And they're weak. It's a weak force, so it's easily broken and reversible. So why? Weak forces are really what govern the chemistry and biochemistry for the most part, right? So ligand binding to its receptor, that's a weak force, and it's reversible. The ligand will eventually go off. So the same thing with the double-stranded DNA. Those weak forces keep those two strands together. They need to be broken, and you don't want to have to input a lot of energy to separate those strands when you're dividing it to make new DNA. Or mRNA, for example. It's universal, occurs between any two atoms that are near each other, and the importance determines the steric complementarity and stabilizes biological macromolecules, including the stacking of DNA. It facilitates binding of polarizable ligands. So the hydrophobic effect refers to the association or folding of nonpolar molecules in the aqueous solution. And it's one of the main factors behind protein folding, protein-protein association, or quaternary structure of proteins. Formation of lipid micelles. Has anybody ever heard the term micelle?
Fat. I mean, like they use like solubilose fats.
Yeah, so detergents, you make micelles with detergents. Detergents will always make micelles. And in fact, there's a critical micelle concentration as you add more and more detergent. There's one concentration where, boom, they'll spontaneously form micelles. But you guys don't know what micelles are?
Little fat.
Yeah. I got pictures. I could draw one, but my art is so bad. But I got pictures coming up. So, binding of steroid hormones to the receptors, and does not arise because of some attractive direct force between the two nonpolar molecules. So they're not directly attracted to each other. They're just excluded from the water because the water wants to decrease the surface area that is exposed to these hydrophobic molecules. It's not a direct attraction between the two molecules. Does that make sense? It's a common misconception. So this is some solubilities of gases in water. So we see that nitrogen is nonpolar, solubility is about 0.018 grams per liter. Oxygen, 0.035. Carbon dioxide is 0.97. And then we get polar gases, it's 900 grams per liter will dissolve in water. And hydrogen sulfide, 1,860 grams per liter. So you can see it's the non-polarity versus the polarity, how they're soluble in water. So the low solubility of hydrophobic solutes can be explained by entropy, right? Like what I was saying. Because water has to be ordered around the nonpolar substances, and that's a loss of entropy. because bulk water has a high entropy, water near hydrophobic solutes is highly ordered, which is unfavorable because it has low entropy, and low entropy is thermodynamically unfavorable. And hydrophobic solutes have a low solubility. So this is what normally happens to a hydrophobic solute. You're going to form a highly ordered lattice structure around that solute. So this represents basically a detergent, like a free fatty acid. You have a carboxylic acid, and then a long chain hydrocarbon, hydrophobic alkyl group. And water is going to be highly ordered in that. And this will still be soluble, but the more and more of this you have in solution, the more these will interact together and form micelles. So, oh, that's one thing I should have mentioned. This is an amphipathic molecule, right? Do you guys know what that is? Amphipathic? Yeah, so it's both, has a hydrophilic portion of the molecule and a hydrophobic portion of the molecule. So it's considered amphipathic, kind of like amphibian. They both live in water and in the air. So amphipathic means it has both, basically. So if you consider amphipathic lipids like a free fatty acid in water, the lipid molecule is going to disperse in the solution. Nonpolar tail of each lipid molecule is surrounded by ordered water molecules, and that decreases the entropy of the system, which is an unfavorable state. You get something like this. You have all this ordered water around each one of these tail groups. It's highly ordered. That might exist for a short person, but soon the nonpolar portions of the amphipathic molecule are going to aggregate together so they're not associated with the water, basically. And the released water molecules end up being more random. There's less surface area of ordered water around those tails. The nonpolar groups are sequestered from water, and the released water molecules increase the entropy. Only the polar head groups are then exposed and make energetically favorable hydrogen bonds with the water. And this isn't a good picture of a micelle. It's just showing you that those three that we had before, if they have these parts interacting together, there's less of a surface area that has to be ordered around those tails. And this is what you'd get if you just put this is like the lattice structure around just like a fat globule with no head groups, just a nonpolar group. You form a really highly ordered setup. But this is a micelle, where you have the head groups on the outside and your hydrophobic core forms in the middle. So the only part interacting with the water are the polar head groups, and they're either going to be ionic or have hydrogen bonding capabilities with the water. And your detergents, your Triton, your Tween, they'll all form these micelles. Any soaps that you use to wash your hands, they form these micelles also, because they both have hydrophilic portions and hydrophobic, so it's a good way to mix and dissolve hydrophobic molecules that wouldn't normally dissolve in water. You can get them into these micelles and wash it away down the sink, off your skin and whatnot. So hydrophobic effect also favors ligand binding. Binding sites and enzymes and receptors are often hydrophobic pockets on enzymes. And if you look at a lot of like drugs that bind to the receptors on enzymes, a lot of those are very hydrophobic type molecules. So they'll end up, and they sterically fit perfectly into the receptor binding sites. Such sites can bind hydrophobic substrates and ligands such as steroid hormones, and many drugs are designed to take advantage of the hydrophobic effect also. I should just stop reading that because I was saying it right before I read it, right? In a different way. I don't know. And this is just a figure from your book showing you that. So if you have some substrate that's hydrophobic, when it interacts with the enzyme, this portion now you don't have to have the water to be ordered, and you've decreased a little bit of the surface area around the enzyme and the substrate. So water also has colligative properties, right? You guys remember that from General Chem? What happens to water as you increase the number of solutes to its boiling point? It drops?
Solubility rises.
No, so boiling point and vapor pressures are related, right? Even though we're not talking about that here. Do you know what happens to the vapor pressure at the boiling point?
It's equal to the pressure.
The atmospheric pressure, right? So, oh man, that's me. Zwitteron. So, this is the way I always used to think about the boiling point and colligative properties of water. So you have a solvent, say it be water, and there's a certain surface area that's going to be allowed to exchange, molecules with the atmosphere. And when this is the amount of vapor that's above your solvent is basically your vapor pressure, right? And the surface area is going to, the amount of surface area you have that's directly exposed is going to affect that vapor pressure. If you start putting solvent or solutes in here, part of that surface is going to be blocked by be contained by the solutes. So that'll decrease your vapor pressure, right? So what are you going to have to do to the energy of this to increase that vapor pressure to equal atmospheric pressure again? Huh? Yeah, you got to increase the temperature right here to increase the vapor pressure. And if you have a lot of solutes blocking the sites here for exchange, you have to increase it higher. So increase solutes is going to increase your boiling point, right? Boiling point elevation. Yeah. Melting point is decreased, right? And then you guys know about osmolarity? Or what's it called?
Osmotic pressure.
Not osmolarity, but what's that? The pressure, osmotic pressure. I can't think. So osmotic pressure you'll find is important, especially in the blood. And all around all your cells, but maintaining blood volume, osmotic pressure, and there's something called the colloid osmotic pressure you'll learn about in the future. So non-colligative properties include your viscosity, surface tension, taste, and color. Depends on the chemical nature of that solute. Cytoplasm of cells are highly concentrated solutions and have a high osmotic pressure, right? Do you remember what osmotic pressure is? That's on the next slide. I don't need to draw it. That's like, so you have pure water. In the center, you have some kind of container with a semi-permeable membrane that'll only let water exchange and not any solutes. So if you have no solutes dissolved in the water, those pressures will be equal. If you have salts in this, water's going to flood in here to a point where the pressure of the air is pushing down on it, and there's enough force here that's forcing it back out. But if you force this down, the force that you apply to that to make it equal again is considered your osmotic pressure. Does that make sense? So, and that's important for cells. Why? Because membranes can be permeable to water. You have a lot of intracellular solutes. You want what's known as the inside of a cell and the outside to be isotonic, or same concentration of solutes. And if they're isotonic, there's no exchange of water. There is, but what goes in comes out at similar rate. In hypertonic solution, the water molecules are going to move out, and that's going to shrink that cell up, take all its water out of it until they're equal concentrations. If you have a hypotonic solution outside the cell, water's going to rush in, and that cell can eventually be burst, right? That's actually one way to lyse cells, like red blood cells for doing flow, if you per se. Ionization of water, so here we get into the acid-base chemistry. So OH bonds are polar, and can dissociate heterolytically, and products are a proton and a hydroxide ion from OH bonds. Dissociation of water is a rapid and reversible process. Most water molecules remain unionized, thus the pure water has a very low electrical conductivity. Resistance of pure water is about 18 mega ohms per centimeter. Do you guys know what ohms is? It's a measure of, how hard it is to get electrical flow through a substance. So very high resistance. Equilibrium strongly at the left of this reaction, so the extent of dissociation depends on the temperature. So protons do not exist free in solution, right? So you have water, they're immediately hydrated to form hydronium or oxonium, and a hydronium ion is a water molecule with a proton associated to one of the lone pairs. Hydronium ions are solvated by nearby molecules, and the covalent hydrogen bonds are interchangeable. So those protons will go from one water to the next, to the next, to the next, in like a constant flood of, proton sharing, basically. Allows for extremely fast mobility of the protons in water via proton hopping. And this is a picture of proton hopping. I'm not a fan of this picture, because I like the electron arrows. This is proton arrows. But it shows you basically the protons are hopping from one hydronium ion, forming a new one, and then they just move around like that in solution nonstop. It's not like they're permanently attached. So ionization of water, concentrations of participating species in an equilibrium process are not independent but related via the equilibrium constant. You guys remember how to make these equilibrium constants? So water will dissociate to hydrogen ions and hydroxide. Equilibrium constant equals the concentration of hydrogen ions and hydroxide ions divided by the concentration of water at equilibrium. And that could be your equilibrium constant. Do you know how that's related to rate constants?
Essentially the same with rate constants. Change the hydrogen ion with
Yeah, so like if you're not at equilibrium, there's a rate for this reverse reaction and a rate for this forward reaction, right? So you'd have like KF times, or that's not what's in brackets, it's like that. And brackets is the H2O, rate of dissociation, I'll make that a D, and then the rate of formation, not H, OH minus, right? And when these two are equal, that's when you're at equilibrium, right? So you have, if rate dissociation equals rate forward, then these are little k's too. The little kd times H2O equals little kf times H plus OH minus. And then we can solve for this, right? And you get KD divided by little f equals divided by the concentration of water. And that, right, is your equilibrium constant, right? Because that's when the forward reaction and the reverse are going to be equal. So that's how you get how the equilibrium constant's related to your rate constants. Your rate constant for dissociation of water divided by the rate constant for the formation of water is basically the KEQ, which is the concentration of hydrogen ions and hydroxide ions divided by the concentration of water. So, and in pure water, your concentration of hydrogen ions and concentration of hydroxide ions is 10 to the minus 7. So you can calculate that from this, right? And you can determine the concentration of water from its density. Do you know how to do that? How do they come up with this 55.5 molar? Anybody know the density of water? Yeah, so 1 milliliter per gram. So in 1 liter there is,
1,000 grams.
yeah, 1,000 grams or 1 kilogram, or 1,000 grams. So to calculate the concentration, we want to go X molar equals 1,000 grams per liter. And then what's the molecular weight of water?
Like 16.
16 plus 2, 18. So we know that the 18 grams of water, there's 1 mole. And so you divide 1,000 by 18. Hopefully that should be like 55. Does anybody got a calculator? I forgot mine. But that's how you calculate the concentration of water from its density. You should end up with 55.5 molar. And then you can solve for the KEQ or KW. So that's how they get that. So, but what is pH? Do you know what the P stands for in pH? Huh? Yeah, negative log, but it got named that because the pH, you can think of it as like the power of hydrogen ions. But still, it confuses a lot of people because what is the pH with a lot of hydrogen ions around? Is that big or small? Small. Yeah, negative log. So lots of hydrogen ions, you have a little pH. Very few hydrogen ions, you've got a large pH. People always mix that up. And I can understand why. I might have too when I was a youth. So pH is defined as the negative logarithm of hydrogen ion concentration. Simplifies the equations because when we're dealing in chemistry, a lot of things are on a logarithmic scale. pH and pOH always add up to 14 because that's the KW, right? Or the pKW. So in a neutral solution, the H equals the hydroxide and the pH is 7. And pH can be negative too, right? So it's a negative log. You can, if you have a high, super high concentration of hydrogen ions, it'll end up with a negative pH. Because I think 1 molar hydrogen chloride is what? Has a pH of what? 1 molar? So hydrogen chloride. So hydrogen chloride pretty much completely it's not supposed to be a capital L. It's going to completely dissociate. So you'll have 1 mole per liter of hydrogen ions. What's the negative log of 1?
0.
Yeah. So if you have a 2 molar hydrogen chloride solution, that'll give you a negative pH, right? So just to show you the negative pHs are around. So, I guess they showed you that here with a 6 molar hydrogen ion concentration would be totally negative, right? So, anyways, this is showing you that it's logarithmic, and hopefully you guys understand that. So 10 to the 0, that's pH is 0, that'd be your 1 molar hydrogen chloride. And then 0.1 would be 1 pH and so forth. And the reverse is true for the OH. As the pH goes down, you have increasing acidity, which equals what? More hydrogen ions. pH goes up, increasing basic, which is less hydrogen ions in solution. And dissociation of weak electrolytes So I gave you the example here, hydrogen chloride's a strong electrolyte. It dissociates pretty much completely. In biochemistry, we work a lot more with weak electrolytes, such as this. Anybody know what this is? Or does it say? No, good, it doesn't say. Not carboxylic, so this would be so there's one methyl group attached to the carboxylic acid group. No, formic is just one carbon. Acetic is two. This is acetic acid. So it dissociates to form acetate and hydronium ions. And its Ka apparently is 1.74 times 10 to the 5. And you can, so the extent of dissociation determined by the acid dissociation constant. So is this part favored or is this favored when it's in solution? If you just take acetic acid and dissolve it in water, are you going to have more protonated acetic acid or dissociated acetic acid?
Protonated.
Yeah, it's heavily protonated because you have a big, 10 to the negative 5. So very little of it dissociates, right? And then we can solve for the amount of hydrogen ions based on the concentration of these times the Ka, because we know the Ka, that's calculated. And you can actually calculate how much of this to add to get a specific pH, right? Or specific concentration of hydrogen ions. And this shows you this equation, right? So, do you guys remember how to solve quadratic equations? You do?
Yeah.
You're not going to need to here. Do you know why we won't use this?
Do you have graphing calculator?
No. But look at this. So it's 0.1, and then we see how much does X end up being?
It's hardly anything.
Yeah, it's really insignificant from the original concentration. So the dissociation is insignificant. So when it's a really weak acid like that, you don't even have to solve this quadratic equation to come up with the pH. So in this example, they solved the quadratic equation and calculated the pH, and they got 2.883. And then the very next slide, our book does it just by ignoring the amount that's lost, the amount that's, subtracting the disassociated from the complete acid. And it's 2.880, so it's insignificant, the changes. But this is still, if you have the Ka, you're going to have X disassociated times X for the hydrogen ion produced. They're going to be equal in concentration, right? Divided by the concentration that you dissolved, and it's X squared times that. You take the square root of both sides and you'll get the concentration of hydrogen ions and acetic or acetate ions in solution. Take the negative log of that and that'll give you the pH. So, do you guys remember those calculations? Was that 2045 that you did that in, or 2046? 2046, I forget. So we might do a couple of those. So, but those are easy, right? And this just shows you pKa measures acidity. So negative log of Ka, so it's basically the same kind of thing. Instead of pH, we have pKa's, and we're going to use that here in a second. So strong acids have small pKa's, or large Ka's, small pKa's. Weak acids have small Ka's, large pKa's. So like phosphoric acid is polyprotic acid, right? So it's got 3 Ka's. And actually this is an important buffer physiologically also, right? Phosphate buffers. So the first dissociation, pKa is 2.14, second 6.86, and the third's at 12.4. So talking about buffers are mixtures of weak acids and their anions or conjugate bases, and they resist changes in pH. And why is that? So if you have a buffer, you want equal concentrations of 50-50 mixture of the acid and its conjugate base. in the solution. So if you start adding more acid, that conjugate base is going to absorb that proton rather than the water. So you won't change the pH so much of the water versus what the conjugate acid is. And if you're adding hydroxide to it, titrating hydroxide in, if you have a 50-50 mix of the acid, it'll give up its proton before the water will. So that's how the buffers work. And that's why, and you'll lose buffering capacity when you're about more plus or minus 1 pH of the buffer capacity. So buffer capacity is the most at the pKa. So if we wanted a buffer for pH 7, looking at, where are we? Looking at phosphate, or looking at these acids, phosphate would be good for a pH, a buffer if we want to keep it about pH 7. Not so good would be glycine, pKa of 9. If we wanted a buffer at pKa 9.6 or so, glycine would be a good buffer. So would carbonate and so forth. So if you're looking for a buffer for a certain pH, you want the pKa to be right around the same range, within 1. And this is just showing you how acetate would work as a buffering solution. If these are equal in concentration, you add a little bit of acid to that, it's going to protonate some of the acetate and absorb that proton versus, and it'll also give that up to a hydroxide if you're adding hydroxide to that solution. And you guys have seen these titration curves before? So you'll have fast movement of the pH as you're adding equivalents of hydroxide to a buffer. Until you reach the buffering range, then it levels out, pH doesn't change until you surpass that buffering range, and then it'll shoot off the charts again. And again, the center point is there where the pH equals the pKa. Weak acids, of course, have different pKa's. This is acetate, acetic acid rather, phosphate, and then ammonia, all different pKa's. So they buffer at different ranges. So the Henderson-Hasselbalch equation, this is a real easy one to derive. I used to have to always derive it because I hated memorizing things. So I would just derive it for my general chem tests every time, because I would always flip this around or whatever. So I'd have to derive it. But basically you solve for the Ka and then you solve for the hydrogen concentration. Take the negative log of both sides, and then you can either use it like this, or you have the pH, remember there's a negative log, pKa here, and then minus the log of that, or you can, if you flip that, that turns into a plus. So real easy equation to remember, and I'm sure there's questions in the book about those to practice, and if you guys want to practice those, and next time we'll go over any questions you have about them, okay? So maintenance of intracellular pH is vital to all cells. Enzyme catalyzed reactions have some optimal pH. And you'll learn later is because pH is going to govern hydrogen bonds between amino acids, so it's going to alter the shape and binding characteristics of those ligand binding sites on enzymes. So there's an optimal pH for certain enzymes. Solubility of polar molecules depends on hydrogen bonding donors and acceptors. Equilibrium between carbon dioxide gas and dissolved bicarb depends on the pH also. So speaking of that, this system is what governs your blood pH, right? Do you know how your body regulates your blood pH?
Oxygen, carbon dioxide?
Not oxygen and carbon dioxide. So carbon dioxide's one way, and then bicarbonate is the other.
So it uses like in the cycle, it's like a little formula that it has like carbonic acid, and then it just...
Yeah, so carbon dioxide, when it's mixed with water, will form carbonic acid, and that'll dissociate to form bicarbonate, right? And then bicarbonate plus a hydrogen ion. And you can write out that equation like this. So you have H2O plus CO2. And that's in equilibrium, will form bicarbonate acid. And then that will break down into hydrogen ions plus bicarbonate. Right? So this is all in equilibrium. So your body can control the concentration of this and this, right? So if you have a high pH, you have low concentration of hydrogen ion. So what do you want to do?
Breathe. Breathe all.
Breathe less, or you can get rid of bicarbonate, right? So if you shift the if you increase the amount of CO2, it'll shift it that way. But also if you decrease the amount of bicarb, you'll shift the reaction this way also and generate more hydrogen ion, right? Do you see how that works? So how does your body get rid of bicarbonate?
I thought it used it in, I think, stomach or something?
Not the stomach. So bicarbonate's the buffer in your blood. Huh? What gets rid of things from your blood for the most part? What are those bubbles in your pee?
Filter it through your kidneys.
Yeah, so you secrete, you kick the bicarbonate through your kidneys, you end up with bicarbonate in your urine. So if you urinate, you see bubbles, that's that bicarbonate turning back, and the carbon dioxide and bubbling out, right? So if you have a alkaline blood, it's going to shift it this way because you're going to kick out the bicarb from your kidneys, shift the equilibrium this way, and start to produce more hydrogen ions. If you have too much acid, you want to blow off more CO2 in your lungs. You'll blow that out of your lungs, and it shifts it back this way and takes the acid out of your blood. So that's how your body will regulate your blood pH. So, and there's actually a Henderson-Hasselbalch equation we could derive that you can calculate this. So if you guys going into CLS, you'll need to do that on your boards, and I'll do that in clinical chem also. So I think I did it last time with the old book, but we'll continue on. So buffer systems in vivo are mainly based on phosphate concentrations in millimolar ranges. Bicarbonate's important, like I just said, in blood plasma. Histidine is efficient buffer at neutral pH. You guys know what histidine is? Probably not, because we haven't covered the amino acids yet. It's one of the amino acids. And buffer systems in vitro are often based on sulfonic acids, cyclic amines such as heaps, pipes, and ches. So, like I was saying, water is involved in reactions. You have an ADP plus a phosphate to make ATP. Forms this phosphoanhydride bond, so you have to remove the water out of that. And that's a dehydration reaction. And that's also how you form, all the polymers are made with these dehydration reactions. So water bound to proteins is essential for function. So this is a protein decorated with water. It's actually the actual crystal structure, and the water stays with that crystal. It's so important to the protein structure. And water bound to proteins is essential for their function also, because it'll coordinate and stabilize these hydrogen bonds. And we'll see in the future when we start doing mechanisms of enzymatic reaction how that'll affect it also. So, what time is it? It's 3 o'clock. So I'll just jump to the amino acids so we can get started on that, like the schedule. So for this, you want to know the basic structure of the amino acids and their base, I'm not going to make you draw them out, but you're going to need to know which ones are basic, acidic, aliphatic, and polar, and aromatic are basically the categories. You'll need to know which ones are which. And we'll learn the structure and properties of peptides, ionization behavior of amino acids and peptides, and the methods to characterize peptides and proteins. So proteins have a lot of different functions. One of the main ones we'll see a lot about is catalysis. Anytime you see something with an ACE on the end, typically indicates it's an enzyme and it has enzymatic properties. Enolase is in the glycolytic pathway. You form an enol. Do you know what an enol is?
It's the negative charge of oxygen, like it's NH2. It's like NH2, and then it's like, oh, it's some.
So an enol, what is it, like?
It's a ketone, right?
Yeah. So there's a keto-enol shift, right, that you've heard of before. So the enol, it gives you a clue right there, the enol. So you have an ene plus an ol, or the alcohol, right? So the ene is what, this? And alcohol. So that's the enol form, the keto form, and it's the same molecule, right?
It's been tautomerized or something like that? Tautomerized?
There's this equilibrium that exists in the molecule where between those. You can have a ketone, and if it's a ketone, it'd be like that, right? Okay, I'm drawing them too close together. So this is the keto form of this molecule, and this would be the enol form. They have the same, molecular structure basically, but you just have a shift of a hydrogen ion from here to here. You form an alcohol from, this will pick up a proton, and then these electrons will have to drop back down there so you can have that. And this gets picked up by some other base. Love my artwork. Right, so this picks up this proton. These electrons will go here. That's keto-enol shift, so that's enolase. You form an enol, and you'll see that in glycolysis. DNA polymerase polymerizes DNA. Again, you have the ACE for transport. We have hemoglobins, important for transport of oxygen. Lactose permease transports lactose across a membrane. Structural proteins include collagen and keratin, and a lot of structural proteins you'll see a lot of novel amino acids and their unusual protein structures with lots of hydrogen bonds and cross links between them. And then motion, where we're converting chemical energy into physical energy or kinetic energy. Myosin and actin are all proteins involved in that. So this is just a picture showing you the keratin in these horns. It's all protein, right? And they're structural proteins. Amino acids are the building blocks of proteins, and they have properties that are well suited to carry out a variety of biological functions. Capacity to polymerize, so that's your peptide bonds. Useful acid-base properties, varied physical properties with all the different R groups, and varied chemical functionality from those R groups. And this is your basic model for an amino acid. You have a carboxylic acid group connected to an alpha carbon that's connected to a hydrogen, some side chain or R group, and then your amine group also. And again, at neutral pHs, these are almost always be Zwitter ions, depending on what the R group is. That could have a charge too, which will throw that off. So, but at neutral pHs, it's amino acids exist as a Zwitter ion. So the alpha carbon always has 4 substitutes, and I just went over those. All except protein have an acidic carboxyl group, a basic amino group, an alpha hydrogen connected to the alpha carbon, and the fourth substituent is the R group, is unique. And in glycine, the fourth substituent is also hydrogen. So because you have basically, except for glycine, 4 different groups connected to one carbon, what is that? Yeah, it's a chiral center, so they're all chiral. And you only have one version of the chirality that exists in nature, basically, so, in our proteins anyway. So organic nomenclature starts from the one end, so you have, that'd be the carboxyl group. The number 2 carbon is the alpha carbon, and if you keep going, such as in lysine, the next would be beta, gamma, delta, and epsilon. I'm terrible with my Greek, so I'm not going to test you on that either. But that's how that goes. And this is lysine. Is this, do you think it's acidic or basic? Yeah, it's a basic amino acid, and it's typically charged at neutral pH. So it has a positive charge. Because again, these are the zwitterion, net neutral, but you have a net positive charge with lysine. So, like I was saying, these chiral centers, which one doesn't have a chiral center? You remember? Not proline. The only one that has a hydrogen for the R group, which is glycine. So, but all the other ones that we find in proteins are L amino acids. So they'll look like these. So. Now, I'm sure you could construct in a lab a full D protein. That might be interesting. I guarantee people have done it. So common amino acids can be placed in 5 basic groups depending on their R substituents. Nonpolar, aliphatic, aromatic, there's 3 in there. Polar, uncharged, so it's going to have an alcohol group most likely on it. Positively charged, which are your basic amino acids, or negatively charged, which are acidic amino acids, right? Acidic amino acids give up their proton, so they'll have a negative charge. Basic amino acids have an amine group typically, which will bind a proton and end up with a positive charge. Does that make sense? And the nonpolar aliphatic groups include glycine, and that's the one that's not a stereoisomer. Alanine just has a methyl group. Proline is an interesting case because it's a ring structure. So that's got a pretty much, it's very restricted to the kind of angles it'll form and structures it'll form. So you find a lot in specific places in proteins. Valine is a branched chain amino acid. Methionine has this sulfur group in between. Isoleucine is like this, another branched chain, but it has the branch on its beta carbon, whereas leucine has its branch on its, let's see, beta. What's the next one? Gamma? Yeah, on the gamma. See, I'm bad at those, so I apologize. Aromatic R groups include phenylalanine, tyrosine, and tryptophan. So, and a lot of these end up being neurotransmitters down the line too. Then serine, threonine, and cysteine. These are all polar uncharged groups. So serine just has an alcohol. Threonine has an alcohol also. Cysteine as a thiol, and these will form disulfide bridges, your cysteines. Glutamine and asparagine are also uncharged polars. So these are amides. And then your positively charged R groups, or your basic R groups, are lysine, arginine, and histidine. And you can see they have the amine groups on lysine and arginine, or this is actually a guanidinio group. And then the histidine is an important, it's typically, because the pKa of this group, this nitrogen here, right here it's in the uncharged form, but it's 6.8, I believe. So it can be either charged or uncharged depending on where it's at in the enzyme. So it's typically important for enzymatic reaction centers. So aspartate and glutamate are your acidic or negatively charged R groups. And if you notice, aspartate and glutamate look a lot like asparagine and glutamine. These are the amides of your acids, basically. So and then there's a whole bunch of uncommon amino acids, and most of them aren't incorporated into the proteins by the ribosomes except for selenocysteine, but they arise via post-translational modifications. So for collagen, for example, you get a lot of hydroxylation. That's important for forming, hydrogen bonds between collagen fibers. And then you have reversible modifications that occur a lot that are important, especially phosphorylation. That's important for signaling and regulation of enzymatic activity. And these are some of the pictures of your modified amino acids found in proteins. 4-hydroxyproline, that's found a lot in collagen. 5-hydroxylysine, it's a major constituent also in collagen. 6N-methyllysine, gamma-carboxyglutamate, and so forth. And then reversible modifications, so you'll see these a lot in proteins: phosphoserine, phosphothreonine, and phosphotyrosine. All of these phosphorylations, these are phosphoesters of the alcohol groups on these side chains. And these will be modified in proteins that affect regulation. And did I mention what types of enzymes add these phosphate groups? Kinases, yeah, or kinases. Might be right. I've always called them kinases. But yeah, kinases do that. Do you know what removes the phosphate groups? Not proteases, phosphatases. So phosphatases will remove these, kinases add them. So N-methylarginine has a methyl group added to the arginine, and then N-acetylysine has an acetyl group. So a lot of proteins get acetylated. Do you guys remember which proteins get acetylated a lot? I don't know if you covered it in core bio. Yeah, so acetylated histones, have you heard of those? Yeah, he has, and Brigida's class taught you about that. So that has to do with how the chromosomes are packaged, whether they're active or not. They'll acetylate and deacetylate the histones that package those chromosomes. And then some proteins get their bases adenylated also, where you have an adenylate ester with them. So do you remember what this is? This part of the molecule here is AMP, right? So ionization, so at acidic pH, low pHs, that carboxyl group is going to be protonated and you won't have a charge, but your amine group will be protonated too, so that'll have a positive charge. So you'll typically have a net positive charge at really low pHs of an amino acid. Neutral pH, the carboxy group's deprotonated, and the amine group is protonated still, so that gives you a net zero charge, and that's what's known as a Zwitter ion. Oh, I erased it, so I can't see if I spelled it right.
I think you missed the I right now.
Yeah, instead of the I, you put an E.
I put an E? See, I am a loser. So at alkaline pH, the amino group then loses its proton and you have a net negative charge because the only thing that's charged is that carboxylic acid. So you get titration curves that look like this. You'll have the buffering region for the carboxylic acid. pKa is around 2.5, 2.3 here for glycine. Continue to titrate it with hydroxide. It'll jump up to the pKa of the amine group, which is around 9.6 or so. And then the average between these two is the PI. Anybody know what PI stands for?
Point of inflection? Point of inflection?
No. So this is the point where there is no net charge at all in that solution. So it's the isoelectric point, or the PI, pH at which there's no net charge. And that's easily calculated with this because there's only 2 pKa's, and it's just halfway in between the two. Okay? So chemical environment affects the pKa values. So alpha carboxy group is much more acidic than in regular carboxylic acids, and that alpha amino group is slightly less basic than in regular amines that you'll see. And amino acids, of course, can act as buffers. The uncharged side chains, such as glycine, have 2 pKa values, and we just saw these. And it can act as a buffer between in those 2 pH regions. It'll act as a buffer. And we'll use some glycine buffers, not us in this class, but in some of the labs otherwise. And this shows you that again. This is rather, shows you the 2 buffering regions, and then the PI again. And Zwitter ions predominate at pH values between the pKa values, and for amino acids without an ionizable side chain, like I was saying, it's very easy to calculate what the PI is. And at this point, the net charge is 0. So, and that's, of course, when it'll be least soluble in water, and it will not migrate an electric field. So what is it when we migrate things in electric fields? Do you know what that's called? Yeah, electrophoresis. So you have to have some sort of net charge for that molecule to migrate in an electrical field during electrophoresis. However, there's one type of electrophoresis that you separate proteins based on their PIs. Does anybody know what that is? That's when it's called isoelectric focusing. So your solid support that you have to do the separation with is really a pH gradient. You'll put the proteins in on one place and they'll migrate until they have no net charge and stop migrating. That's how you can separate proteins based on their PI. Then you can take that and transfer it to a regular gel and separate based on size. So ionizable side chains can also be titrated. Titration curves are now more complex. pKa values are discernible if the 2 pKa values are more than 2 pH units apart, so otherwise it'll look like a big mess. And why is the side chain pKa so much higher? You look like this. So this is for histidine. So pKa1 is about 1.82. The pKa2, which is this nitrogen, so below the 6.0, it's protonated, and above it's deprotonated. So your pKa2 is there, or pKar for the R group. pKa2 is 9.17. So the PI then, if you look for which species doesn't have a charge, you take the 2 pKa's that surround that to find out what the PI is for that, rather than the other 2. So, and that's what this describes, is you identify the species that carries no net zero charge, identify the pKa value that defines the acidic strength, and the pKa that defines the base strength of this Zwitter ion, and take the average of the 2 pKa values. So, and to do that, you're going to use this pKa, or the pKr, and the pK2, and the middle between those two is 7.59, if you do the math. So formation of peptides. So peptides are small condensation products of amino acids, and they're small compared to other proteins, less than 10,000 Daltons. So they're small polymers of amino acids, and they're formed by this condensation dehydration reaction that forms the amide or the peptide bond. So, and then I guess we can stop here because it's late, right?
It's about 3:30.
Huh?
It's about 3:30.
Yeah. So, and don't we stop at 3:40, I guess? So I don't want to get too far into this, and then because this is kind of a new section, right? So then we don't meet again till next Wednesday, right? So, and if you guys can bring questions to me, I'm in the lab all the time. I'm going to be in the lab basically Monday through Thursday from 9 to 7 for the most part. Huh?
Tuesday through Thursday?
Monday through Thursday. Yeah.
Okay, so lab's open Monday?
Yeah.
Even though it's a holiday?
Not this Monday.
Oh, okay.
So, yeah. Sorry. Were you hoping? Or?